Good afternoon, everyone. I want to start off by just thanking the organizing committee, especially Dr. Lauren Koske, for this very kind invite and allowing us to present some of the work or interest that we have at Gilead with HIV cure. And also thank you to the audience for sticking out this long. I'll try to make it as brief as possible. It just makes sense for us as an antiviral company to be now pushing the frontiers to the next stage, that is looking at HIV cure. So I just wanted to present some of our interests in this area. So most of my talk is going to be giving an overview of latency and viral persistence, some insights into or thoughts into strategies for eradication, as well as sort of briefly touching on some of our ideas and interests in this arena. So to sort of give an introduction in terms of describing viral persistence and latency, it's sort of appropriate to sort of think about this in the context of viral dynamics and decay in patients that are on suppressive HART. And so when HART is initiated, what you essentially have is a stopping of ongoing replication. So there's no newly infected cells coming into the system. And therefore, by looking at this viral decay, you can then begin to get some sense of the size, the relative size, as well as the decay dynamics of these reservoirs that carry or harbor latent virus. And so I do want to say up front, all of this information that I'm going to be discussing has actually come out from some very nice, elegant work done by several lead labs in the field, including Dr. Fauci, Bob Silicano, John Coffin, et cetera, numerous studies from some elegant work. So as soon as HART is initiated, what you see is a very sharp, rapid decline in plasma viremia within as little as 1 to 2 weeks. And this essentially comes from the first phase of decay, which is represented by these activated CD4 T cells that are productively spitting out virus. Following this is a slightly slower phase of decay, which of course comes from a reservoir that has a slightly longer half-life, and this is suspected to be the macrophage reservoir. So these are not necessarily latent, they're actively producing virus, but have a slightly longer half-life. The second phase essentially brings this viral load now to levels that are essentially undetectable or at the level of detection by most commercial methods of detection, which is 50 copies per mil. And so with these pieces of information, and also the fact that patients that are compliant with their medications can maintain these suppressive viral loads for very long periods of time. And so given these pieces of information and extrapolating this second phase of decay, it was thought that it should be possible to clear off all of this virus or the infection in as little as 2 to 3 years. However, in spite of the type of combination of therapy or the duration of this well-suppressed aviremic phase, as soon as the HART or the medication is stopped, there's a rapid rebound in virus in these patients. And so it became pretty evident that despite absence of detectable plasma viremia, there is persistent virus still present in these individuals, and it is obviously coming from some kind of long-lived reservoir. Of course, then there was the development of much more sensitive methods of detecting virus, single copy assays. This is primarily from the efforts of John Coffin and Frank Maldarelli's lab, Sarah Palmer, where they're able to detect down to a single copy of RNA per mil of plasma. And from these studies, it became clear that in these patients that do not show or were not initially showing any detectable virus, you could actually now, with these more sensitive methods, show that there is a long-lived persistent viremia that still exists. And this essentially identified a third phase of decay, a third phase of decay where the half-life was estimated to be about 44 months. And so now, extrapolating this phase of decay, it became evident that clearly it's going to take many more years, about 73 years, to clear this 1 million latently infected resting CD4 T cells. Of course, this is estimated from resting CD4 T cells, so not even accounting for all of the various latent reservoirs that may be present. So the next question obviously was, what are the sources of this persistent stable virus? And obviously the first is to consider are these cellular reservoirs. The most extensively studied is the memory CD4 positive T cells. It's estimated there's about 1 million latently infected resting CD4 T cells in an individual. This is an approximation. And in macrophages, these are not necessarily latent. They do actively produce virus, but are slightly more long-lived compared to the activated T cells. And then, of course, there's evidence now emerging that additional cell types, including hematopoietic progenitor cells, even naive or dendritic cells, may harbor low percentages of these latently infected cells. The second important factor for us to consider is the possibility that there is ongoing replication. So we believe, because of the very low levels of viral replication, or very low levels of viremia, or that there isn't any ongoing replication, or that ongoing viral replication is well suppressed by current HART regimens. So to address this, some of the studies that were done was intensification of HART, so adding on a fourth agent to the already suppressive HART regimen. For instance, adding raltegravir to an already 3-drug combination, et cetera. And none of these studies essentially showed any evidence of any decrease in this residual viremia. A hallmark of viral replication is also evolution of the virus. So you see the emergence of mutations, accumulating mutations. Some elegant studies, again, done by the lab of Bob Silicano, showed that when they sequence these plasma populations of virus, they do not see any detectable evolution of the virus when they take samples over a long period of time. So these lines of evidence suggest that they may not be ongoing rounds of replications. However, we cannot completely discount the fact that there may be some very low levels that are below the limit of detection by these methods. So it cannot be completely discounted. Nonetheless, what this does suggest is that the predominant amount or the predominant levels of plasma viremia, the persistent viremia, clearly cannot be coming from ongoing rounds of replication. It has to be coming from some cellular reservoir that is long-lived. The anatomical reservoirs, CNS, GU tract, where they may be slightly compromised to drug penetration, can also offer sites where the virus can be more long-lived. And of course, homeostatic proliferation, the maintenance of the reservoir, essentially these memory T cells. So in a way, when we think about latency, it actually is, in some sense, some byproduct or a biology of the immune system itself, that is the maintenance of these memory T cells. However, these also happen to be the sites where HIV happens to live. So clearly, reservoirs impose a clear barrier to HIV eradication strategies. So then you ask the question, is cure possible? Well, perhaps maybe a different way of asking the question is, what type of cure may be, what type of cure we may consider as potentially possible down the road. So there's two approaches to this that are being considered. One is the idea of sterilizing cure. So this sort of reflects the infectious diseases model, where the idea is that treatment with the drug would result in complete eradication of all infected cells. So in an HIV individual, the idea is that the approach will then eliminate all infected cells in that individual. So your viral RNA copies should essentially be undetectable in plasma as well as tissue samples. The other approach is the idea of functional cure. And here it's more based on a cancer model, where the idea is treatment with the agent will result in remission. The idea being that you can have long-term, patients can expect to have a long-term healthy life in the absence of HART. And here, it's not necessarily that you completely deplete the virus, but bring it down to a level that is low enough where the immune system or the body is able to control it. And there are examples of both the functional and sterilizing cure. In the one case, there's the Berlin patient, the cancer patient that underwent bone marrow transplant, and here cells were received from a donor that is CCR5 defective. So the patient had to go off HART medication because of complications with the cancer medication that he was going through. However, what was interesting was that when he was taken off heart, there was no rebound in viremia. And in fact, now almost more than 3 years since stopping heart and post-transplant, this patient does not show any evidence of or any indication of plasma viremia, virus either in the plasma or tissue samples. In the case of functional cure, the elite controllers are a very good example here. They comprise a unique group of HIV-infected individuals where virus is present, but at very low levels, below 50 copies per mil. And so the body, so in this case, the patients are able to control that low level of virus through their immune system. So obviously, approaches like this are not really realistic for HIV-positive patients, bone marrow transplant. However, what these examples allow us is, gives us good examples where we can now begin to explore or ask the questions how and why virus is controlled in these individuals, and therefore we can identify targets to then go after to try and recapitulate these situations. So something like an elite controller perhaps may be more realistic as opposed to completely eliminating all of the infected cells. And so then thinking about such approaches, one of the well-characterized reservoirs for HIV has been memory CD4 T cells. And so we can then begin to think of what approaches might then we apply to these systems where we can tickle the virus to come out. And so if you think about it, some of the agents that have been tested or approaches that have been considered, for instance, are HDAC inhibitors. So this is through chromatin remodeling. HDAC inhibitors have been explored primarily for cancer indications. So what you see is treatment results in certain genes that are being overexpressed, certain genes that are being shut down. But in the case of HIV, you do see an upregulation of the virus. So epigenetic control of HIV. Other approaches, PKC activators. Here, you're working through transcriptional factors, upregulating transcriptional factors that then the HIV genes would be sensitive to. PTEF-B, as you've already heard from previous talks, some of Chung-Su's studies have shown the role of PTEF-B in transcriptional activation of HIV involving TAT. So there are multiple ways by which HIV can be tickled to come out. However, we have to keep in mind that none of these are really selective approaches that only activates HIV without disturbing any other genes. So they're sort of generic approaches. The ideal agent would be one that will activate latent virus without inducing global T cell activation or activation of the immune cells. Additionally, what you also want is that when the virus is activated, you want these latently infected cells then to die out, either by the cytopathic effects of the virus or through immune clearance. However, this aspect of this eradication strategy is still at an assumption stage. It's thought that the cells would be cleared out, but it's yet to be proven that this will really happen. So we kind of have to think about what level of activation is necessary and how broad of a signal that we need to apply. And so talking a little bit more about these activation agents, as I mentioned, PKC activators. A very well-described example is prostratin. Another one is bryostatin. Both of these are natural products. They primarily work through signaling through the PKC protein kinase C. One of the end signaling molecule, or one of the end factors here is the activation of NF-kappa B, so a transcriptional factor that HIV responds to. So methods that do upregulate transcriptional factors can then result in activation of HIV. Another class of agents are inhibitors of the histone deacetylases. These are enzymes that are zinc metalloenzymes. They catalyze the removal of acetyl groups. Their target proteins are not necessarily just histones. They also act on numerous other proteins, importantly also transcription factors. And so here the mechanism, as most of you already know, the acetylation status of the chromatin dictates the ability to express genes from, or controls gene expression. So in the deacetylated state, the chromatin is a more condensed conformation. You have less access to transcriptional factors and therefore not as much expression of the viral protein, viral gene. When treated with an HDAC inhibitor, you have inhibition of the enzymes, and therefore it shifts to the right. You have more acetylation, an open chromatin structure, and therefore more permissive to transcriptional factors, and you have gene upregulation. Saha is one of the most extensively studied HDAC inhibitor that has been used for activation of HIV. This is an FDA-approved drug for CTCL, cutaneous T-cell lymphoma. And it's been shown to activate latent HIV in resting CD4 T cells in numerous studies as I've listed over here. And this is one such example. And this is essentially cells that have been taken from HIV-positive individuals, CD4-positive T cells taken from individuals, so about 15 different patients. And compared to PHA, which is a positive control here, you can see Saha also is able to activate and produce virus outgrowth in these cells. What's very nice about HDAC inhibitors, unlike with prostatins, is that it does not induce T cell activation. There are several HDAC inhibitors now currently in clinical development. And I wanted to just point out that, however, there's only 2 so far that have received FDA approval, and both of these, again, are for cancer studies for cutaneous T-cell lymphoma. But there are several others that are still in various phases of clinical development. Also, with the HDAC inhibitors, you can see that they vary in their class specificity. The hydroxamate-based compounds, similar to Saha, panobinostat, belinostat, these tend to be more pan-HDAC, that is inhibiting a broad number of HDACs. So HDACs essentially are 11 isozymes classified into 4 groups. The pan-HDAC inhibitors inhibit multiple enzymes from these classes. Some of these, for instance, romidepsin, this is a cyclic peptide. It is more selective for class 1. These benzamide-based compounds, the methylgene and entinostat, are also class 1 selective and only inhibit a select set of HDAC inhibitors. There was a nice study actually done by David Margolis in collaboration with Merck that had shown that in patients in cells, when they treated with different flavors of these HDAC inhibitors, the pan-HDAC inhibitors seem to have the highest success in activating virus. So what's of interest for us is now some of these HDAC inhibitors actually moving into clinical trials for HIV eradication strategies. And there's 2 of such studies that are currently enrolling patients, one here in the US, headed by David Margolis, and another one in Australia by Sharon Lewin. For us at Gilead, definitely we have interest in all of these agents that have been shown to activate virus. For the HDAC inhibitors, we do have our own internal program, not necessarily specifically for cure, but for other indications. And we also have a very broad collection of compounds that are both pan-HDACs as well as class-specific. PKC activators, we have a collaboration currently ongoing with Professor Paul Winter at Stanford. And therefore, this gives us access to synthetic analogs of prostatin as well as bryostatins, so PKC activators. And then, of course, we have now just starting into a collaboration with Professor Chung Zhu, looking at molecules that might perturb these PTFB complexes and shift them into the more active complex that can then upregulate HIV. And of course, we also have setups for looking into HTS campaigns using primary cell-based HIV latency assays. And of course, the field is, of course, thinking of it as not just looking at individual agents, because if you think about it, there's the most amount of HIV activation happens when you have global T-cell activation. So therefore, you have multiple signaling pathways that are fired up. And therefore, it just makes sense then by combining multiple of these agents, you could potentially then have more effective or more robust upregulation of virus. And so in trying to make all of this happen, of course, the key component for us is the development of assays. And cell-based assays is very key here. And there's different flavors that we're looking at. Cell lines, of course, are very easy, amenable to high throughput. However, these do not represent quiescence in the true sense. And they're not really representing latent infection. So yes, they're still valuable. However, they do not really capture all of the factors that may be driving latency. Virus outgrowth assays using cells from HIV positive patients. Clearly, these are not very high throughput type of assays, but nonetheless, what's valuable about this is that we can now get cells that are actually coming from HIV positive patients that have been on long-term HART. And so by working with leukopaks, they now give us the capability to do large-scale assays. The key assays that we are really going after are primary cell-based assays. So these really allow us to capture latency in cells that are HIV-specific. And at the same time, they also offer reasonably high throughput compared to what's achievable with these patient cells. So there's several flavors of the virus, several flavors of primary cell-based assays that are currently out there. Most of you may know about the Silicano, the assay developed by the Silicano group. These are using CD4 T cells transduced with BCL2. They're infected to transduced, allowed to quiesce, then infected with virus. The Planeus and Bosque study has shown this nice system using CD4 T cells, naive CD4 T cells. These are activated and primed under conditions that allow the generation of central memory phenotype cells. These are then infected with reporter viruses. And Sharon Lewin's group at University of Monash has this chemokine model of latency, where again, they take resting CD4 T cells, treated with these chemokines, and then infect. So again, the cells are not activated. They still are able to capture latency. So multiple flavors of the assay, the main point here being that these are primary CD4 T cells, which is very critical. They all capture latent infection, again, very important. The cells are quiescent. They're not in an activated state. They carry an infection. They carry integrated DNA, which is quantifiable. We can also see that these are latent infections in the sense that viral message is very low. You're not expressing high levels of virus. However, they do respond to reactivation stimuli and express high levels of virus when treated with the compound. In addition to this, we also have highly sensitive quantitative PCR-based assays. And this is just to show that we can go down to copy numbers as low as 10 to 20 copies of RNA per mil. So this is about 10 particles of virus per mil. So gives us gives us a very useful tool as we move into animal studies. So at Gilead, of course, as a pharmaceutical company, we operate on these projects as a multidisciplinary team. There's several teams that are involved in sort of making all of this happen. So at the start, we have assays that allow us to identify activators. We, of course, have the known compounds, internal projects that allow us to identify agents, collaborations, as I've already described, and also HDS campaigns that then allow us to identify new molecules that may identify new targets. These hits from these are then put through an elaborate series of assays for characterizing, understanding mechanism of action, and they're also validated in these T cells that are derived from patients for confirmation. They then go through another series of QCs, eventually moving into these animal studies for determining PK/PD and toxicology. Once we've identified a set of compounds that have an acceptable therapeutic window, we can then move these into proof of concept studies in Rhesus macaques. And this is just sort of to show what the method would involve. So the most critical component of this test of concept study is the establishment of a cohort of latently infected Rhesus macaques. So here, the combination of drugs, the choice of the animal, and the virus, et cetera, all play into this establishing this cohort. So what we want is a very effective antiviral drug regimen that allows in these Rhesus macaques to have a very effective long-term suppressive HART regimen. We then come in with the agents that we've identified, do the reactivation study. Of course, here it's critical to determine what dose, frequency, and route of administration. And then lastly, look for the outcome. And this is done through several markers, viral markers that are plasma virus, as well as cell-associated RNA and DNA. And of course, lastly, we can, of course, stop the HART medication, and then we would look at the rebound kinetics in the treated versus untreated animal. So in summary, in terms of considerations for eradication strategies, clearly latently infected resting memory CD4 T cells represent just one component that has been the most well-studied, of course, being a low-lying fruit. However, this is a very good start point nonetheless. Multiple mechanisms, of course, drive latency, and therefore combination approaches are likely to be most effective. Ongoing replication, this is still very much in debate. Do we need to have additional agents put on board? Is heart intensification required as we do these eradication strategies? And then will virus activation lead to cell death? This is very key, because if you're not achieving that, then these eradication strategies are really not going to achieve what we're trying to get. Better understanding of the biology as well as latency, better understanding of the biology and the immune system, and development of effective animal models. And so with that, I'd like to thank our really dynamic team. This is a group of people from various disciplines: biology, medicinal chemistry, drug metabolism, toxicology, and IP. And of course, our most important, valuable contributors, collaborators, Jeff Lifson at the NCI, Koen van Rompuy, Paul Luciu, and Tom North from UC Davis for some of the animal studies. Tom now, of course, is at Emory. Paul Wender at Stanford for the PKC activators, and Chang Zhu for collaborations on the PTFB project. Thank you, and I'll take any questions.
I'll have the first question. So,
Sure.
You mentioned there are several compounds, Saha, HMBA, post-stratin, that are somewhat effective in inducing latency, reactivating latency.
Right.
Are you doing any rationalized design to improve efficacy and perhaps reduce toxicity, in addition to your HTS campaign?
Right. So the collaboration with Paul Wender is where we're looking at the prostratin analogs. And then we have our internal program for the HDAC inhibitor, so we have different flavors other than Saha.
Hi. It's very timely to end the sessions with HIV and this idea of cure. I just want to put it in perspective. I think you did a nice balanced job. I happen to be on the side that feels that eradication is virtually impossible unless you do it in the early stages. You were right. You have virus in the brain, you have virus in the GU tract, testes, you have virus in the kidney, you have virus in the pancreas. Those people who think just getting it out of the immune system is going to eliminate this virus and make it a real cure, to me, are naive. So I just go on record, everyone knows this. Functional cure, we're talking about something else. But a functional cure, which is what we think the Berlin patient is, not a sterilizing cure, but a functional cure in cancer means these people are no longer on anti-cancer drugs. A functional cure in HIV infection says you will probably need to give them drugs. Because when you stop all the treatment, what you thought is you've brought it out of all the white cells in the body. You won't get rid of it, my own opinion, in these cells, long-lived cells of the brain, or long-lived cells in endothelial cells, unless they're shed, which will restart the infection again. And I just hope your long group of collaborators at least appreciate this when you're going into it. I think for a model, and Paul, this is important for you, I think you should use a sooty mangabe. They don't get sick. They have a very high viral load. Give them heart, bring it way down. They will have virus in the brain. They will have virus in other and see what happens. Right. One of the other problems is, who is going to have the courage, after giving these patients all these drugs to activate the virus, to stop heart? Because these long-lived cells may take 4 or 5 years. So again, these are the kinds of questions you should raise. What is your milestone? And can you reach it to any benefit, which to me would be, can you reduce the number of drugs that you have to give? So those kinds of things that you would hope to get. Of course, we all would like to see a situation, at least in my own group, where we induce the immune system to take over, then you won't need the drugs.
Right, right. No, you bring up all very good points. So we don't want to go into this without being naive about it. Absolutely. We realize that this is really a pretty high goal to achieve. But the animal studies will really give us some valuable data. And again, the choice of the animal, the choice of the virus, what combinations, and then the suppressive drug regimens, and clearly establishing that you have very good suppression, long-term suppression in these animals, identifying and studying the reservoirs that are developed is in itself going to be a challenge, I think. So there are different flavors of the animal systems out there already to begin with. So I think, but we have to start somewhere. And so if we can at least deplete something and have some evidence that we've depleted something, that in itself is a important milestone. So I think we're kind of trying to look at it as, a step at a time.
There's a question in the back.
I think Dr. Levy already half asked my question because he said that it would be preferable to have the immune system take over because that's the more natural situation where we do see near sterilizing immunity. So in a sense, wouldn't it be a reasonable point to, instead of looking for a absolute eradicator, which seems to be perhaps impossible or very close to impossible, to search, to use this post, or not post, but low-level infection after or during heart as a time for vaccine. So as a development for a vaccine, which would take, which would lead the body to take care of the few remaining cells.
Right. Yeah, the idea of functional cure is.
Leonid, that's what they're doing. They're trying to give this eradication approach and then do a post-infection immunization. Get some proteins to immunize so you get, after all, you need the immune system to take care of the virus.
Right, yeah.
Well, you think the drugs will, but you want the immune. My colleagues think that if the immune system is there, if the virus doesn't kill the cells, the immune system will. That takes a lot of faith.
Yes.
Because the virus replicating, and those of us working in cell culture know, you can get lots of cells that can continue to produce HIV, never die. I mean, they will eventually die, but yeah. So you would then induce an immune response through some immunization. And I'm sure you're considering that as well. That's where they're trying to, but you hit on exactly the way I feel. They really got to boost the immune system if you're going to stop the drugs.
Yeah, and it doesn't have to be completely, I mean, what I said as in taking off, the idea of completely taking off heart is eventually, say, 20, 30 years down the road maybe. But for now, if we can even show that the rebound kinetics have changed, that is a start. And then you can then go to the next stage of saying, okay, we've depleted something. What is the evidence of what we have depleted? And then go to the next step. So maybe even decrease heart number of regimens that you need to have. I mean, the idea is to have lesser and lesser, or not to have long-term combination therapy and therefore not have the associated side effects. Yeah.
All right, so with those thought-provoking questions and answers, we end our scientific program. So please join me in thanking all the speakers for their wonderful talks. And of course, the organizers, Lauren Koskoy, and also the wonderful staff members of SAND for organizing this event. So what's going to follow is going to have 3 panelists telling us what they think the program will become in the future. And after that, I was told there's a reception in the lobby or in downstairs with alcohol to further stimulate our scientific discussion. All right.
Introduce yourselves before we lose everyone. All right.
Okay. If you can all just wait about 5 minutes, because we will have a reception afterwards. We want to share some ideas that came up during a lunchtime session, which was closed, and included just a few faculty members and postdocs to come up with ideas to take this meeting to the next step. What are some of the resources that this community could use? What are some of the ideas that have been generated by this meeting? So we have 3 postdocs here to take us forward, and I'll let them introduce themselves.
Well, good afternoon. I'm Simone Zompi. I work in Eva Harris lab, and I've been working mostly on the memory B-cell response in mouse, and now translating into human to dengue virus infection. And I've been summarizing some of the talk we had during lunch around translating findings from the bench to the field. So I will tell you about that.
Okay, I'm Molly O'Hanley, and I'm from Eva Harris's lab as well, and she talked about my work today, so you've heard about that. And Marta, who can introduce herself in a second, and I are going to talk about a little brainstorming session we had about how to keep the momentum going from today's symposium and sort of a Bay Area virology group sort of idea.
I'm Marta Gallia. I work in Britt Glanzinger's lab, and I work on the mechanism of RNA degradation in herpes viruses, and I'll be talking with Molly about creating a virology community in the Bay Area.
We'll go first, I guess. We have a couple
Go ahead.
I'll just come up here. So yeah, Marta and I are going to kind of share the table. We just have a couple slides. So the brainstorming session that we had was basically about how we feel like there's a need to strengthen collaborations amongst local labs. So a lot of us have a lot of collaborations all over the place, but wouldn't it be nice if we had something more local to kind of come back to? So the basic ideas are what other labs doing, how are they doing it, and how can we help each other out? So just to kind of outline what was talked about. The biggest thing was maybe some sort of central website. So if there's a Bay Area virology group or something where we have a website listing labs in the area that are doing virology research. So at the UC campuses, Stanford, Lawrence Livermore, Torrey, other campuses, and also industry, obviously, as well. Sort of a research description of what people do, perhaps reagents and protocols that they might have to share. A big topic was also computational resources. So there's a need for shared server space, perhaps, as well as sharing of code and scripts for people that do bioinformatic work. It would also be nice to share facilities, or at least to know what exists. So different types of BSL facilities that different places might have, as well as flow cytometry, mass spec, sequencing, and other types of sort of core facilities that all the campuses or that companies might be able to provide. Or also Lawrence Livermore seems to have a lot of assay development, so different things that people might be able to apply to their question. Let Marta take it from here.
I guess we also discussed how that may be complemented by having a listserv where people can ask direct questions about reagents. And there are scientific networking sites that we could use to start a website without having to maintain one ourselves, and some that came up were ResearchGate and Copernicus, which are kind of like Facebook for scientists, or at least that's how they present themselves. A corollary to that would be to continue the momentum started by the symposium. So there was a general sense that we would like the symposium to become a regular event, although perhaps we should discuss what kind of format people want, and also what kind of recurrence. So we talked about having an annual symposium, but also perhaps having more frequent meetings, having a virology supergroups in the Bay Area with a rotating location, or perhaps a seminar series. That would, of course, require a certain amount of commitment in terms of travel between campuses. So we weren't sure whether that would be feasible. And also, there's a lot of industry working on infectious disease in the area, and perhaps we could use this forum to boost interaction between industry and academia. People, as usual, would really like to have social events where scientists can meet, academic scientists can meet industry contacts and talk about working in industry, but also there are lots of grants that are aimed at specific industry academia collaborations, and having contacts in industry would, of course, open up a lot of opportunities for people to apply for those grants and start collaboration with product development more in mind. So, of course, it's very important for any of this to actually come into being, that the community is interested in having some sort of greater forum for virology in the Bay Area. And Laurent talked about how he is planning to use the collection of emails that he's put together for the symposium to contact people about their interest in developing regular meetings and regular resources. And of course, the bigger the endeavor, the more sponsorship and funding will be required to actually make it happen. But that's going to be more possible depending on how committed and interested people are going to be to develop it. And I guess we were thinking if you guys had suggestions or comments that we'd love to have your input on what you would like to see happen and start.
It would be nice to have more young scientists present and postdocs and students.
That's definitely something we talked about, was what kind of format, and maybe there's a place for more younger scientists to present, and places where it's better to keep it to PIs and such. But that's something that is up for discussion, I guess. So.
Just a couple of comments. Plant viruses, we've learned a lot. They're big problems and all that. So I'd like to see a little more representation of plant viruses, and I'm from UC Davis, so there are a lot of colleagues. But then just one or two other comments. To add to your facilities capabilities, here we have the California National Primate Center, which is a unique resource, and that was alluded to here and all that. And we'd like to get more involved in asking questions about mechanisms of viral pathogenesis and actually testing some of the hypotheses that are coming out, for instance, from these virus-host protein interactions. And the Rhesus macaque genome, the sequence was published about 4 years ago, so you can very quickly move from human to macaque. Mm-hmm.
Go for it. Yeah. Questions on the first? No? Oh, okay. So, so I was at this roundtable where we've been discussing how to translate science from the bench to the field, whether it's in developing country, but also around here in the Bay. And so one of the major issues that everybody brought up, it's sustainability. Whether you are setting up a new rapid assay at San Francisco General Hospital or an epidemiological surveillance in Zimbabwe, how to make it sustainable. And one of the, these are a few ideas that the people who were there at the round table brought up. And one of the main thing is do a needs assessment first and work from the bottom up. So go there and ask what do they need and work from there. Of course, it has to be tailored to each country. Each country is different, and even sometimes each area, even in one city, can be different. So the second point, which is also linked to sustainability, it's training. And one of the main point that came out also was the best thing to do is to train on site, train local people and train on site as much as possible. And also don't forget that science, it's not only pipetting, but there is how to look for founds, how to write proposals, how to write manuscript once they have done something. So when you think about training, also think about these missing links. One main local leader also, it can be very important and can help sustainability in the long term. Quality control, it's important for, especially if you are thinking about setting up a site for a future clinical trial. And again, here you have to think about all these little things that we may forget when we are just at the bench pipetting, and these are a good biostatistician. It's important to have it since the beginning because it can change the way you set up your trial, your experiment, your cohort, or your study. Informatics, data collection, all these are things that can be set up in the field and again, by training locally. And finally, translational science at large. How do we translate the academic discovery to the big pharma? The usual, what we usually see is a person from the academia who will do a startup, and then a bigger pharmaceutical company will buy the startup. But is this the only model? Are there other ways to do it? So if you have any ideas, that would be great. And also, one of the points that has been brought up is how to circumvent these very long trials and FDA approvals, especially when you have people that are really in need of urgent care, and you're like, well, we know this compound works, but we have 10 years to go before it gets approved. So any good idea, either will open the floor to discussion. I gave too many ideas.
I have a comment to offer on the last point that you made. FDA's single requirement is safety, safety, and safety, and nothing else. As far as the efficacy is considered, it's the secondary consideration. So on all FDA committees, the advisory committees like professors sit on it, ultimately it's the bureaucrats who run the FDA, they make the decision. Mm-hmm. So the delays that you're talking of are almost inevitable, number one. Number two is that even for compassionate use or any other experimental use, you still need what is called IND, investigational new drug. Mm-hmm. And without the FDA approval for IND, even compassionate use is not possible. So I just want to make that point to postdocs or young students who do not know modus operandi of FDA.
Mm-hmm. Thank you.
Any more questions? No?
I just wanted to comment on the first part where it demanded about some suggestions. So I do understand that for most of the talks, it will be the PIs and the young scientists or postdocs may not get time to present. And so if the poster session could be extended, because that's where the postdocs are representing themselves and they talk about the techniques in more detail, so that would give a good understanding to the audiences or other virologists to understand what's going on in each lab. So if that could be extended. Thank you.
I fully agree with that. And so I just, before we close, I know you don't want to hear anyone talking anymore, so that...
There's one more question.
One more.
So I want to ask, do you intend to somehow disseminate those talks and posters? This is a way to encourage further collaboration and creating those connections that you were talking about.
Yeah. So,
maybe put them on a website or something.
That's what's planned, and every talk has been, videotaped, and they will be put on a web. So they will be there, they will be available, and we'll send an email on where to look for those. And also what I wanted to say is look out for your emails, because I will be sending, not tomorrow, but within the week, just an email, because I will ask feedbacks. And we would like to know what exactly you want, in terms of improvement that we can afford, in terms of speakers, lengths of the speakers, and everything we talked about. So I would like to get all the feedback possible to make it even better next year. So From that, I think we can close, and there is a reception, so there is drinks and some finger food. The poster is supposed to be still on the board, so people can still walk, through the poster for an hour, and, Thank you very much for everything, and, yeah.
Last, thank you to Laura.
So, welcome. Thank you.